Okay. So welcome everybody. We're so excited to have you join us today to meet Raquel Miralles, who is our new Science and Research Director. And she is incredibly well placed to be in this position and help us move forward with the work that we're doing in SCN8A and really working to advance progress and get us closer to better care, treatments, and cures and quality of life. So we're thrilled that she's on our team. It's still relatively new, just about 8 weeks now. So ish, something like that.
Yeah.
Uh, so we're going to hand it over to Raquel, who's going to tell you a little bit about herself, but also walk a little bit through the scientific research and funding process so that people have a sense of some of the stuff that's on the horizon for us and how we've invested time and energy in the past into advancing the field. So I'm going to hand it over to you, Raquel.
All right. Thank you, Gabby. Hi, everyone. Nice to meet you. Oh, I'm spotlighted. Awesome. Um, oh my gosh, my desk chair just broke. I'm so sorry. What a bad time for that to happen. Um, but so nice to meet all of you if I haven't met you before. My name is Raquel Morales, and I have previously worked in the SCN8A community, but I'm really excited to be in this new role. So I've included, oh, this is there we go. A little bit about me. So I did a PhD in neuroscience at the University of Virginia. I worked in Dr. Manoj Patel's lab. You might know him because we just had a webinar with him last week. And I studied the basic mechanisms of SCN8A, and I helped with the gene editing study that they presented on last week, which is why I was able to answer some of those questions. For me, science communication is a really big priority. It's always been a really uh, high priority for me because of my background. I grew up in a rural area and I grew up to a father who's Hispanic. And so, from someone from sort of an underrepresented background, we didn't get a lot of exposure to science when I was really young. And so, being able to bridge science to people from lots of different communities is really important to me. I'm also really invested in patient advocacy. So I did some previous work with the Epilepsy Foundation of Virginia, and I'm a family member to someone with epilepsy. So these are all just little things about me. Um, and if you have thoughts or questions or anything, feel free to share them in the chat, or this is a pretty small meeting. So if you want to ask me directly, that's also totally fine. Um, but I'm going to talk a little bit about understanding scientific research and sort of the process uh, that goes into science, because I think a lot of us will see the outputs of science, but not sort of what goes into it. And that's a really, really big part of how we keep science moving and everything moving to help CNA. So one of the big questions is sort of who does basic science research. So the big three categories are academic labs, biotech and pharma, and these things called CROs, which are contract research organizations in academic labs. They're typically funded by government grants, with some grants from biotech and the pharma industry, or some nonprofit grants. They're often driven by publications, which is fancy word for just papers. So if you see a research paper on SCNA, that's the main um, output for these academic labs. And oftentimes, academic labs are collaborative because oftentimes they're really willing or required by their funding mechanism to share their results. So they're usually really willing to help other labs out. With biotech and pharma, they're definitely commercially focused and oftentimes funded by investors and their revenue from their products. They have big goals to bring new therapies to market, and they're very regulated and often have some limited data sharing, just because of the nature of finding new therapies and bringing them to market. With contract research organizations, these are for-profit companies that will support other organizations. Some biotech and pharma or academic labs might hire a contract research organization to do something specific, and they're often paid by their clients to execute some sort of research project under a very specific timeline. Um, but I have the most familiarity with academic labs, and also that is a big focus of, I think, the research that we're hoping to drive forward, is that basic research through academic labs. That's a lot of what I'll be talking about in this presentation. So another important thing to consider is how long does scientific research take? Um, so new research can take a while and it requires funding, like I mentioned in the last slide. Um, one experiment can take anywhere from a few days to a few months or more. There are some people whose experiments it probably has taken over a year, which is a little crazy, but um, they want to make sure that the experiments that they do are really, really uh, robust and comprehensive. And a scientific paper can often take years to generate. So if you read a paper that's, 15 pages or so, oftentimes it takes whoever's doing it a really, really long time to generate it. So um, scientific research is really important, but it definitely takes a lot of drive to get to the finish line. And so, like I mentioned, scientific research is expensive. So this is a little look into the funding scope. So to fund a PhD student, federal funding is usually 35 to 50K for 2-ish years. To fund a postdoctoral scholar, so basically once a PhD student graduates, they often stay in a different research lab before they're trained enough to become a professor. But usually they're funded about $50,000 to $70,000 a year for about 3 years. And then to run your own lab, the government kind of gold standard grant is around $500,000 a year for 4 to 5 years, which, as you can see, that adds up to a lot of money. Also, just as a terminology note, scientists often use the term PI, which just stands for principal investigator, when referring to independent researchers with their own labs. So if you hear or read anything about a PI, that's what it means, not a private investigator or anything like that. When I first started in science, that's what I thought it meant. Um, but it's an important sort of terminology thing to think about. And so, in terms of the academic funding timeline, how people get funding, uh, a lot of times the first step is gathering their ideas and gathering some supporting data. So the big part about getting a grant is that scientists need to do preliminary experiments to even start writing a grant. Because without sort of preliminary experiments that set up the backbone behind a grant, there's no proof that any of their scientific ideas are even possible. And so that's a really big part of getting academic funding and getting those big grants for $500,000 a year. Of course, a big part of getting a grant is writing a grant. Grants are really long. Even grants for graduate students can be 50 plus pages in total. And so all of those sorts of grants include background, significance, research design, a really comprehensive budget and timeline. So it takes a long time to kind of comprise those all together. And then once an academic gets to the point where they're submitting their grant, it usually takes about 6 months to hear back about grant funding. And a pretty comprehensive team of other scientists review grants and then they score them. And only about the top 10% of grants is funded. So it's really, really hard to get a grant. And those grants are what we call peer reviewed because a team of other scientists reviews them. Grants are often resubmitted. So only the top 10% is often funded. And so um, oftentimes the people who don't get in that 10%, they go in for resubmission. And so usually scientists don't get a grant on their first try, and the next 6 months or so are spent resubmitting that grant application with all the changes that the team of other scientists, those peer reviewers, gave them. And so overall, this is a really long timeline. It's often 2+ years before, a grant that's conceptualized gets to actually being funded. And that's a pretty long time because, as you saw in the previous slide, scientific experiments are really expensive. Um, and another important aspect is when we're thinking about that peer review, thinking about how these proposals are evaluated. How do they pick the best science to give those really big grants to? Um, so these are the 5 main categories from the National Institutes of Health in the United States. So the first one is significance. So it's how important is the proposed research to the field. The second one is the investigator. So does the scientist that is applying for the grant have the qualifications and experience to carry out whatever research they're proposing? Number three is innovation. So does the research proposal that the scientist is proposing take a new approach to the field? Is it kind of a big word that is used in science is novel. So is it novel? Is it exciting? And that just means is it new? Is it innovative? Number four is the approach. So is the research plan ambitious yet feasible? Whoever is judging grants wants to make sure that their money is actually going to a place that's going to make a difference. And so they want to make sure that whatever is proposed is actually possible and the research methods are appropriate. And number five is does the scientist have proper support from their university, their colleagues? So do they have the proper sort of materials, the proper um, expertise if they're doing something out of their wheelhouse. Um, and these categories all come together to be what is evaluated on for those grant proposals. And no one category is more important than the other. All of these are weighted equally. It's just the order that they're listed in the grant review process. But this is just important to think about when we're thinking about how science gets done. This is what scientists think about all the time when they're trying to decide what project they want to take on. They have to consider all these things because in order to do the experiments, they have to get funding for them. And so a big question is how can our community support SCN8A research? And how can we help advance science for the topics that we care about? And so this is something that I'm hoping to take a large role on with the Alliance and, making sure that we're advancing patient and family priorities. So talk a little bit about the role of SCN8A Alliance, or at least what we want it to be with all of this. We aim to bring families together. So we've worked really hard, or I guess not me, but the team before me, and I'm hoping to help with all of this. It really helped with aligning all the community priorities on SCN8A and bridging the gap to the scientific community. So they've done a lot of that work previously with things like the research roadmap, which I'll talk to you about a little bit later, um, and also just amplifying family voices through collecting data. Um, Another big part of our role is convening researchers. Uh, so bringing researchers together leads to a lot of collaboration, which advances all of our priorities faster. And also convening researchers means that we can have a space to inform professionals of the SCN8A community priorities. And then organizing some community-focused research. So we want to combine our individual efforts and make something bigger possible and get community-focused research on patient priorities so that we can shape research to the directions that we really, really care about. And we want to make the biggest impact that we can with whatever funds we have. So some ways that we have done this previously have been early investigator funding. So that's giving some money to scientists earlier in their career because that helps them a lot with securing larger amounts later. Like I mentioned earlier, it takes some sort of baseline experiments to get a scientist to acquire large funding. And so that early investigator funding can help them bridge the gap to those bigger grants. We also want to bridge some gaps for current researchers. So things like federal funding cuts can impact SCN8A researchers and might be able to bridge a gap in some really important projects that we want to stay alive. And we also want to bring some new researchers into SCN8A. So this is what we're hoping, we're hoping to bring in some new people, and especially folks with expertise in non-seizure outcomes, because a big part of those sort of criteria for funding is the expertise of the person. And so we want people with expertise in some of those areas that we care about to be brought into the SCN8A field. And so what we've done previously in each of these categories, previously we've funded some really incredible people. So JC Wagner, Chris Mackinson, Guy Hellman, Chad Fraser, and all are still working in SCN8A or other rare genetic diseases. JC Wagner and Chris Mackinson have their own independent labs focused on SCN8A, doing some really awesome work. And so it's kind of a sign of impact. So we, give them a small amount of money a while ago, and it has sort of blossomed into a lot more funding for SCN8A and a lot more research on SCN8A. um, bridging some gaps for current researchers. Previously, the Alliance funded Rosie Button-Session, who worked a lot with the R1872W mouse model, which if you were at the webinar for gene editing, that is the mouse model that allowed those researchers to do that gene editing work. And so if this hadn't happened, then maybe that wouldn't have been possible. So that is a really big impact from a pretty relatively small grant compared to those really big ones we talked about earlier. Um, and then funding for researchers new to SCN8A. Maya Jha, who some of you probably aren't familiar with, but she is an MD PhD student, and she was kind of brought into the SCN8A field through working with the Alliance, and she's continuing work on SCN8A as a PhD student by working on loss of function variants, and I think a little bit on how current medications affect SCN8A, which is really exciting and important. And so with all of this, those small funding pockets are kind of they're called seed funding. And we aim to organize research through our community, but we want the biggest impact for our dollars. So this is kind of why we're interested in seed funding. This is sort of small project to get the small funding to get a project started. And it allows researchers to get enough data to propose a bigger grant. Um, but still, it's a competitive grant application process. So it's still comprehensive, but it's a bit smaller in terms of monetary scale, but really, really high in impact. And so I want to talk a little bit more about big impacts from small funding with kind of more direct examples. Example number one, in 2016, the Alliance/AES funded a $45,000 postdoctoral fellowship for Chris Mackinson, who stayed in SCN8A research and now has his own lab at Columbia with independent funding that totals almost $2 million, which is to be distributed over the course of 5 years, which is incredible. It's an incredible impact. Example number two, this is not something that we funded, but this is just to give sort of a conceptualization of how this works. In 2023, the Patel Lab at UVA received a $50,000 grant from the Ivy Biomedical Innovation Fund, and they designed base editors for SCN8A and are establishing a biotech company. So these are kind of examples of things that if we were able to kind of pool together this amount of money, we could be doing this kind of stuff. So we can get in on the ground floor on things like base editing, is the hope. And so we really hope to organize something like this through the SCN8A community to fund family priorities in research, which we sort of outline in the research roadmap. And so this is a little bit talking about the research roadmap itself, but this is sort of what sort of things we want to fund and how the areas that we want to advance research in. So of course, we have the research roadmap areas. So transformative therapeutics, that's things like the gene editing, CRISPR, antisense oligonucleotides, all those transformative therapies are really important. Non-seizure outcomes. We want to get more research in both basic and clinical areas for non-seizure outcomes. And those are really, really critical and identified as one of the top patient and caregiver priorities. Improving current therapeutics, so things like current anti-seizure medicines, understanding why current anti-seizure medicines like Keppra maybe don't work as well on SCN8A, discovering and applying biomarkers. So that'll help us find new biomarkers that eventually could hopefully be used to generate better treatments for SCN8A. And then getting a better understanding of like the whole brain and the whole body, because obviously, as we know, there's not just one system involved with SCN8A. It's not just the central nervous system, and so we need to get a better understanding of how everything is working together. And so this is sort of how we're thinking it might be funded. So if we say, 50 birthday fundraisers lead to $1,000 each, that's a patient priority, a funded patient priority research grant. And so we're hoping that this sort of thing will be funded through community funding because ultimately we're a lot stronger together as a community, and we're hoping to do this sort of thing to advance patient priorities. And the way that we will look at these grants is through our Science Advisory Council. So our Global Science Advisory Council, which is coming soon, would review and select what should be funded based on our criteria. Um, and I'm going to go into that a little bit on the next slide. Um, So these are the categories I kind of went over earlier, the significance, investigator, innovation, approach, and environment. But one that's really important that needs to be included in anything that we review is patient priorities. And we want to make sure all the research that we push forward is aligning with the collective patient and family priorities, um, because that is at the head of our mission. And so we really want to start gathering community funded research, um, and making that possible so we can advance the priorities from the research roadmap. And so that's all I have. So I would love to answer any questions. And I'll stop screen sharing so that hopefully we can have a more informal conversation. I'm happy to answer any questions you all have, either if you want to talk in the chat or if you want to talk out loud. I'm happy to just talk about science, talk about the scientific process, or just about anything you want to talk about. Great. Awesome.
So I see a question in the chat. Do you see that? Will a parent be able to choose where their money goes, as in what research study?
Yeah, so I think the idea will be that so when we're doing this, we are gonna basically send out a request for proposals. And we'll be able to get like a whole bunch of proposals into sort of a pool. And basically the idea would be that the scientific advisory board would review those and select the best ones. And so depending on how much funding we have, we might be able to fund certain ones. But the main thing is we want to be able to fund the research that's the most likely to actually get done. So it needs to be done by someone who has, the right expertise, the right environment, and the right ideas. And so it might be hard to exactly direct where personal funds go, just because we need to make sure that the funds are going to someone somewhere that can push something forward. Um, I don't know if you also have thoughts on that, Gabi.
Yeah,
Um,
no, I think I'll also add that I think what's going to happen as well is that we'll try to move forward on things where there's the most movement and the most kind of energy behind. So for example, if the was saying, we really want to learn more about GI issues in SCN8A, the community could come together and say, we really want to fund somebody to do that work. And then families can go forward and say, if you wanted to do a personal fundraiser, and we start an effort, and then all the people who want to be a part of something like that can say, we'd like to raise money for this kind of a pot of funds to go to this. So, and then if we get enough funding, so if we have a $50,000 target, people can do their fundraisers under that umbrella and say, we want to raise money for GI challenges, research. So, this takes some work to get set up and allocate things in the right way, but we really, our priorities are going to be moving forward on what the community has told us is most important to them. And I don't know, Raquel, if you want to talk about the 1451 mouse as another example, potentially
yeah.
something so of families who are really motivated.
Yeah, absolutely. So I think basically looking at, like Gabi said, if we could get a pool of a certain amount of money, we could direct it towards a specific thing. So as an example, the some of the families who have a child with a mutation in 1451 are really excited about creating a mouse model of that variant. And so they've sort of pulled together some funds and are working with a researcher, and we've kind of done that through like talking to some of the community, and figuring out who might be the best for this work, and it'll end up being a collaboration, which is awesome. But because they really wanted that research done, they were able to direct their funding there. So I think it is possible, depending on the amount of funding that is um, sort of pooled to focus on one specific area. But the main thing I think is we want to be able to get a bunch of proposals from different researchers so that we can assess which one is the most likely to drive the field forward. And when I say we, I don't mean just me. I mean, oh, sorry.
Lo siento tanto,
And somebody else have
pero
another
tengo que
question?
patear y tengo que orinar justo ahora.
Dice, is that you? Are you okay? I think that was maybe a miss.
Sorry.
That's
Oh
okay.
no, you're okay. That's okay.
Yeah, let us know if we can support you at all. Yeah, and I think the important thing when we're talking about funding is, the general funding is also really important for organizations that allows us to, do things like hire Raquel and hold scientific meetings. And so like there's different ways that we'll need to keep the Alliance running and funded, but we also understand that there's really important projects, and some of those we are eager to help support and move forward if we know that there is a movement of families who are also ready to get behind and help us do that. And then we would use this Science Advisory Council to help us get the best research, most high impact research funded, as Raquel was saying. And there also might be cases with things like GI issues. Oh, sorry, it's got the hiccups. I don't know if you guys can hear it. It's very loud, but there might be cases, as for example with GI, where the issue is not just for SCN8A. It might be that, lots of other disorders have similar challenges. And so we could pool together money as a community and say, these three disorders, if we come together with $100,000, we could really push something forward at a faster rate, because we'll still get answers about SCN8A if we have a pool of SCN8A in there, but it would allow a much larger kind of scope to be covered in research. So I think there's a lot of ways we can go at this. And as we continue to build out the Science Advisory Council, which is soon to come, we'll be updating you guys on what that's going to look like. And if you have people you want to suggest, get involved in helping us decide about research granting. So stay tuned.
Yeah. And I think specifically, so for what we're hoping for things like non-seizure outcomes like GI dysfunction, is to bring some new researchers into the SCN8A field. So like that's that last category I was talking about, because it's such an important aspect of research that the lab and the person who's doing the research is well equipped to do it. So like I did my PhD in neuroscience, and I only know how to work with neurons in a lab. And so if someone was to fund me for doing GI work, I wouldn't even know where to start. And it wouldn't be a very efficient use of funds to give a neuroscientist money to do GI work or to do like hypotonia work, just because that's not where their expertise lies. And so that's sort of the thought behind getting people to review it, because they review people and they look at, do they have the expertise, and they kind of score it. And it's like a more objective criteria. Yeah, so the big challenge with sort of cross disease studies, I'm sure I should read the chat out loud. Sorry. Yeah, 2A was not able to be studied with Minaj's particular study, just because of how his base editor works, because it's designed for a specific gene. Um, it is a bit challenging in basic science to do some cross disease translations, but it's definitely possible. And so it sometimes requires different models. And Minaj just doesn't happen to have any models of 2A. And he could get them if he really wanted to get them and work on 2A, and maybe he will. Um, but we're hoping to find some allies that are really excited about cross disease research, if we end up going that direction with this specific project. Um, and yeah, Minaj he doesn't have the right tools in his tool belt, basically. But he can get the tools, and I think he does want to. But right now, he just doesn't have them.
Yeah. So I got a good question here, which is, some folks want to understand better what a neuroscientist does. What kind of work does a neuroscientist do, and what kind of things might you investigate?
Yeah, absolutely. So in the day-to-day, what a neuroscientist does is we go to a lab. And so labs look really different based on what type of lab they are. And so some labs are biochemistry labs, which is basically fancy words for um, saying that they put like liquids into other liquids. And uh, it's sort of like the scientists that you see in the movies, but that wear the lab coat with the little pipette and it makes fun colors. It's a little bit more like that. Some scientists, some neuroscientists are they do a lot of behavior. So that usually involves, and a big core part of neuroscience often involves working with animals. So a lot of people who work on epilepsy related disorders work with mice, just because they're the animals that are the easiest to observe seizures and sort of behavior in. So a lot of epilepsy related researchers work in mice. And so people who do behavior will study how the mice behave. And so there's lots of different sort of tests to see, like, does the mouse have a movement disorder? Does the mouse have anxiety? Does the mouse have depression? Does the mouse have issues with grasping and muscle tone? Um, some people specialize in that. And then some people specialize in sort of how the brain functions. And so they'll do things with a whole lot of electronics because the brain is all about electricity. And so they'll get a whole electrical setup to figure out how the brain works. A lot of times in an animal that is alive, which is really cool, or in brain cells that are still alive. And so that's sort of the three main types of neuroscientists. I can tell you that I'm the last one, um, and that's the one I definitely know the most about. But I know about all the different types. But that's kind of the day to day of what the scientists do in the lab. And a lot of what a scientist does also is reading papers, so reading all the publications and staying up to date in the field. Um, and then a lot of writing and presenting and going to conferences, because one of the most important parts of science, at least to me, is communicating to other scientists, but ideally also people who are not scientists.
Yeah, no, that was a very comprehensive answer. I appreciate that. I think it helps me to understand too, all the different, and there's so many different kinds of labs that are each studying different kinds of topics within each. And, there are many labs that are studying SCN8A, and a lot of them have different focuses. So they're very complementary, because the SCN8A community has been pretty well linked um, over the last decade or so. We held our first scientific meeting in 2015, and that was, people had said that was kind of really a catalyst to get these people together and talking and sharing and thinking. And so I think as they've developed their labs, each person kind of has a little different piece of the pie, and there's less overlap. And that's a really, really critically important part of how we as patient advocacy organizations work with scientists and researchers, is help them understand, what our concerns are, but also who's doing what in the field already and how they can work together and how they can maybe align so that, like this 1451 mouse project, potentially it could be that one lab does the work to create the mouse and another lab does the work to really determine what all of the to test it to see what it looks like, what is the phenotype, how does it present, those kinds of things. I'm probably saying that wrong, Raquel, but you can.
Yeah, no, actually, I was going to bring that up, just because basically one of the things that we can do as a patient advocacy group is bring labs together. And so, like I mentioned, there are some neuroscientists that spend a lot of time with the mouse doing behavior, and there are some that do all the electrical stuff. So with that 1451 mouse, there's going to be one lab that does all of the mouse behavior and figures out how the mice are acting if they have things like cognitive impairment and developmental delay, and then another lab that does all of the sort of underlying things that are happening in the brain cells. themselves and the underlying things that are happening in the sodium channels themselves. So that I think is going to be one of our roles in directing research is linking people together because nowadays neuroscientists are really hyper specialized. Neuroscientists and other types of scientists, because science is so specific. Labs tend to focus on one really specific thing and they're really, really good at it. And so a big part of our role, since we have a disorder that is so, it has so many features to it, we want to make sure that everything that is a clinical aspect of SCN8A is addressed by the researchers. And so we might need a bunch of different researchers, and hopefully we can be the ones that bring them together.
Yeah, and one of the follow-up questions is, in their labs in the research, do they use anti-seizure medications in their mice models to learn things? And like we know there's a cohort that really does better with sodium channel blockers. There's some that do better, with calcium channel blockers. So how do they study that in labs?
Yeah, absolutely. So the answer, frankly, is that they don't really. Um, which is really unfortunate, but that's an example of something that we could fund. That's something that is unlikely to be funded through the NIH through like really large funding mechanisms, just because um, it for basic science doesn't spur a lot of things, but for patient priorities, it's really, really important. So that is one of the areas from our roadmap, and that's an example of one of the things that we could move forward if we had sort of community funding. Because a lot of times when scientists are studying SCN8A, they're studying mice that are on no sodium channel blockers, which is not a great representation of the patient population. That's something that I would constantly bring up in my old lab, but obviously it didn't work. But it is a really, really important aspect because that affects a lot of SCN8A. It affects development. It affects probably a lot of the non-seizure outcomes. And we don't fully understand what those things do to the progression of SCN8A. So that's in the research roadmap, and that's definitely something that we could push forward if we had community support behind it.
There's a few instances of it, right? I mean, like Jen Wong and Andrew S. So a very small amount
Yeah.
do, but also, you might want to talk about how some labs also work with drug companies to do early testing. Yeah.
Yeah, they do. And yeah, I think mostly I was talking about polypharmacy. So basically when patients are on multiple drugs, not a lot of labs do that. And I know that almost all SCN8A patients are on multiple drugs. That's kind of what I was referring to, which I realized I didn't make clear.
Hmm.
Um,
Okay.
some people will do like one drug or two drug, or not even two. Some people will do one drug. Oftentimes it's a very commonly indicated one, like carbamazepine or that kind of thing. Um, but very few people do multiple. Um, but yeah, some labs do get funding to do initial studies for pharma companies. And so they'll be the ones initially screening drugs on mice a lot of the time, and seeing how usually the outcome measure there is seizures and how many seizures the mice have, and that sort of thing. Um, and it isn't a really it's a similar funding number to some of the funding that we provided previously for researchers. So if we were interested in things like that, that's definitely something that we could organize behind.
Okay. So one of the things I wanted to talk with you about, so you've had experience, you've been in the SCN8 community for a while. People may recognize you from scientific meetings, the Acute Syndrome Foundation meetings that you attended for several years. And we certainly know you from years and years of scientific meetings, like the American Epilepsy Society and those kinds of things. And then you've also done work with the Epilepsy Foundation in Virginia. So, you've got some background around engaging with the community, around working in epilepsy. And of course, there's a portion of SCN8A families who don't have epilepsy, but still are impacted by SCN8A. So there's certainly a part of the community that we will be working with as well. But What is your vision about how you hope to work with the SCN8A community and engage with them?
Yeah, I'm really hoping to have more accessible resources and just being a more accessible sort of scientist in the field. I feel like it's sometimes hard to access scientists and ask them questions and that sort of thing. So that's a really important priority to me. And making sure that research and science are a little bit more understandable to families. So like things that we're doing right now, I've just sort of settled into the role. So I did a lot of travel at the beginning and then I moved immediately after. So it took a little while to settle in. Um, like Gabi mentioned, I've been here for like 8 weeks or so. And so it's been a lot going on. But something that we're developing right now is like a pinned Instagram post on some of the SCN8A science basics. So I can pull that up right now.
You guys get a sneak peek. This is
A
not
little
yet
sneak peek.
released, so we welcome feedback from you on what's understandable and what's not. But
Um,
and as you're pulling that up, there's a question about how much an average seed grant usually needs to be.
yeah, so I would say probably around 50,000 is how much they usually are. And so that's because basically, like I showed in one of the slides earlier, a PhD student usually costs about $35,000 a year. And so let's say you're funding a PhD student to do some work for a year, and then you're also funding the materials to do the work, is basically what it would be. Um, because all the science reagent or also reagents is a big word, I realize that. The science materials, which is like reagents, which are like the liquids that they kind of combine together and that kind of stuff, they cost a lot of money, unfortunately. And so they there needs to be some money for supplies in there. So usually about $50,000 funds a PhD student who is someone who's working towards their degree, but they have a lot of experience in science already. Um, and then it'll fund those supplies to make sure that they can have everything they need to do the experiments they want to do.
And you're getting a very good rate when you do that because you're also getting the expertise of their mentors who are very often very established people in the field. So it's not just the science they're doing, it's the mentorship of the people who they work with as well. And then
Exactly.
one thing I'll throw in there that we can talk about after this is also that there's clinical research, which is different than, lab research. And that would be things like if we wanted to actually have clinicians evaluating people and doing, blood tests and those kinds of things to learn more. So that's a little bit different, but we'll get into that down the road when
Awesome.
those arise. So yeah, exciting to share.
Yeah. And I can share this. I realize it's coming up as individual pictures. So I'm not sure if I can switch between them. I think I can, hopefully. Um, but we've got a very hopefully accessible overview. And so it starts out by talking about, what is SCN8A, and that SCN8A produces sodium channels. And hopefully let me know if any of this is not accessible, but that the DNA that codes that it is SCN8A, because SCN8A is a gene, creates components that make up sodium channels. Um, and then it's hard to open all these up.
Oh, you have to do it individually.
Yes, I do. I don't know why it's not.
Can you share
Uh,
it in Canva and then click through?
I can't share it without sharing all of the comments.
Ah, okay. Sorry, I kind of put you on the spot with this. But yeah,
Oh, it's
as
all
we're
good.
moving along, people, feel free to give us feedback if there's something that's not clear on this.
Um, yeah, so this is just talking about that sodium channels like NAD 1.6, which is what SCNA codes for, um, are located in brain cells. So it's a diagram of what a neuron looks like, with where the sodium channels are. So they're right here. Um, they're not just right there, but that's kind of where they're concentrated. And that's a really important part of the neuron. Um, and that the sodium channels are a passageway for sodium to enter the neuron. Um, and then a little slide talking about, I don't know how fast these ones are switching on you guys' end, about why sodium channels are important. because neurons transmit information through electrochemical signals, and that the sodium channels help, generate and spread the signals throughout the neuron, and that's really important for proper functioning. And then a little slide talking about SCN8A variants and mutations. So it's showing DNA and a change in DNA, which is a single base mutation, and that the bases they make up what DNA is. And if any of this is not useful, let me know because I really appreciate all of your feedback. So this is that a base change can lead to a change in the amino acid, which is the building blocks of proteins, basically. And that change in the amino acid is what leads to the sort of different SCN8A protein, and consequently, its different function. Um, and variants can lead to gain or loss of function, but also mixed function, which we have in there. And this is just basically an overview of gain of function. Excess sodium can pass through the channel, which results in increased neuron activity. And then we have a slide, and this is all hopefully going to go on Instagram as something you can swipe through and look at, and be pinned at the top of our page so we can look at it as many times as we want. And so these are things that you all are probably familiar with, but the common clinical signs and symptoms of gain of function. And then we've got a similar sort of thing for loss of function, which is when less sodium can pass through the channel, which often results in decreased neuron activity. And then the clinical features of the common clinical features of loss of function. So I don't know if this is cool to see, but this is the kind of stuff that we're hoping to develop. We're hoping to develop another similar set of diagrams like this comparing SCN2A and SCN8A. We're also hoping to do some plain language research article summaries for the latest SCN8A research. I realize I'm saying we, and that's because there's someone who's helping me out with some of this. There's a science communications intern who's awesome, and she's been working with me on some of this stuff. And so we're hoping to develop all those kinds of things and lots of resources for families, and then just be really accessible to families to answer any science questions.
So any feedback on that is welcome. Did people find that accessible? Were you able to understand what it was? Does something like that help you to better understand how SCN8A variants impact? So feel free to follow up in a message if you don't have feedback right now, or when we put it out, you're welcome to share. We want to learn from you about what works. We got another question about whether there are any medications, and whether there's any kind of novel medications being used to treat seizures in SCN8A.
Not that I know. I think that's something that we're interested in. So we're hoping to enroll in some like AI studies for drug repurposing. So one thing that's really growing in the field is using different drugs that are indicated for other diseases, and a lot of times they'll help with a different one. And so that's something that we're looking at and interested in, and hopefully we'll move forward with that, and that will hopefully give us some info on what drugs we could possibly repurpose. But a lot of the sort of new studies with drugs in SCN8A are done through pharma companies, so I think they're a little more secretive. And so I don't have the information on those.
Yeah.
But
There's some
when we do
labs that
have them,
are, yeah. There are some labs that are doing things like looking at hypertension drugs or, other kinds of classes of drugs that exist that could potentially be helpful in SCN8A, helping with seizures, helping with behaviors, helping with all kinds of other things. But um, like Raquel said, as of right now, a lot of the research that have been done, that might be trial and error in a lab that comes to light. But what we are hoping we can do is use, artificial intelligence to help us look through and see how all of these different drugs work in the body, and if there are some that might act on SCN8A that could help reduce or increase the flow of sodium into the channel. So I think there's the potential for that. We had the webinar last week with a lab who's doing some work in, base editing, which is actually changing the genetic code, right? There are antisense oligonucleotides. I always have a hard time with that word, but which are, that's like a precision medicine where they go in and they kind of often it's with RNA and they just kind of rework it so that anyway, it's there
yeah,
are lots of different things in the works, but nothing is really ready to go.
yeah, I have a pretty cool diagram on antisense I also struggle with that word. Oligonucleotides that could be useful that I can always send out. But yeah, like you were saying, it's RNA. The main problem with SCN8A is I think on the last webinar day I mentioned like a Goldilocks problem is it's hard to not have too much or too little because if you miss the mark of the exact amount even a little bit it causes some issues. Um, and so SCN8A is a hard one for those antisense oligonucleotides. Um, but also, in reference to that gene editing webinar that Gabby was talking about, we made a document for families to post questions. So I'm going to just drop that link in the chat if anyone wants to ask questions about the gene editing at all. That document is in the chat now. And feel free to ask any questions. It's at the very bottom. There's a space for additional questions. And if you ask them in a language that's not English, we'll try to use a translation service.
Yeah.
But yeah, happy to answer any questions there to the best of our ability in a pretty plain language summary sort of way.
Yeah. Does anybody have any other science-y kind of questions? You're welcome to ask. And then I think we'll have, a lot more opportunities to interact with Raquel and ask her questions and lots more kind of posts that she's going to be creating, a lot more, webinar type things where we bring you the latest research, a lot more kind of breaking down what new publications are sharing. So, we're just really grateful that we have Raquel to help us really support you all. Her job is to help us advance the research and get us closer to better care and treatments and quality of life for our kids, but also to be here to support all of us as family members who need help understanding what's going on and can help us think about how we can have impact as well. So please feel free to reach out to us, to her, to any of us if you have questions or need anything. Um, yeah, I think also we'll probably be having Raquel, accessible via our Facebook group, and have her in there so that, she could be available to help answer some questions as well, or support, you know, um, our families as we're kind of going through all of this. So, um, and I think one of the really important things as well that Raquel brought up is that, this patient-centered idea of funding research and making research move forward with that. Um, and it really is going to be based on, we heard from 175 families as we were doing preparations for our research roadmap meeting, and those 5 priorities that Raquel shared, those were from hearing from all of you who participated in that. That was, roughly a fifth of the world's population with SCN8A participated in that. And that's our guide. We will continue to come back to the community to ask for insights and inputs. But, from that, we have priorities that are important for gain of function families, for loss of function families, for different gene variant groups, for different kinds of non-seizure challenges that families want to see more research into. So that's going to be our guiding light. And that paper is soon going to be published about the research roadmap. And we're working on a strategic plan from that roadmap and from all of our engagement with the community, and not just the families, but also with the researchers and the clinicians and the pharmaceutical companies working in the space. And really thinking about how do we proceed over the next 5 years to get and go after the most exciting and promising ways to advance the science of SCN8A. So I think that's really what we want to share with all of you, and that, we are really going to be looking to do a lot more science funding. And that's going to be a critical way for our community to come together. And Raquel mentioned, birthday fundraisers. That's a great way to get involved. Lemonade stands. If you have creative ideas about, if you know somebody who has a art gallery and might want to host something, if you know somebody who works at a big company and might want to help see if they could do, a day to donate to a certain company. There's lots of ways. Just think about your network around you and the resources that you have around you, because we certainly know that individual families alone are not going to be able to supplement this and fund this work. It's got to be rallying our community around us and really thinking about how we build a broad group of people who want to help us get closer to these better treatments. Jada, you're muted.
I just wanted to quickly add that while he wasn't able to make it today, that Raquel is working very closely with Dr. Hammer, meeting weekly, and that one of the real strengths of our community has been the kind of commitment and effort that he has made over the last 12 years, from discovering the gene to starting the registry to doing basic research. So Michael is someone who does both basic research and clinical research and publishes. So his time, while he commits a huge amount of time to the family meetings, he is very consumed. He has students and whatever. So there's no loss of continuity, and there's built-in really good coordination, so that in some ways Raquel is already working with Dr. Hammer to help get more of the data that's in the registry communicated back to families in regular ways and on our website. And as Gabi mentioned, in response to queries that come up in our Facebook group, as in our family meetings, very often Michael will pull some data from the registry in answer to someone's question. So Raquel will be leveraging, the knowledge and expertise that Michael has, but really building on that and expanding. And there's a strong and committed partnership there. So this is not a breakdown in any way of the central role that Dr. Hammer has and continues to play in the Alliance work.
Yeah, absolutely. And like Jada was saying, I've been working really closely with Dr. Hammer, and Gabi and Jada, I can be the first to tell you that sometimes in those weekly meetings, we've just kind of gone off about science, just chatting about, SNA science and ways to move it forward and that sort of thing. So our relationship is really good. And one of the coolest parts is that I talked about the different types of neuroscientists, and we're different types, which means that we cover different areas and we're stronger together, basically, because he knows a ton of things that I don't know, and I know some things that he doesn't know, which is awesome. So it's a great thing to have him, and yeah, he does amazing things for the community. I'm glad to work with him.
Yeah. And I'll add one last piece, which is this idea that, as a global SCN8A community, we're a very small community, but we have a lot of impact working together. So a lot of this stuff we're partnering with our global leaders. So we have Celeste here, who's from Portugal, and there's a group of 10 other patient advocacy groups around the world that are part of this SCN8A Global Leaders Alliance. And we'll be working with them on this Science Advisory Council to get proposals and funding for different things. And so they'll each be involved in whatever parts they want to be in as well. And so it's really pooling the community together globally. We don't want to take anything from what they're doing. They have leadership in their countries and their communities with their folks. And it's really us creating a path for anybody who wants to fund science research to come in, make sure they can get the best science funded, make sure they can get the most for their money, and move the field forward. And although TCSF is not a part of the Global Leaders Alliance, we are also going to be working closely with them so that they can align on science funding research. So, this is really for the full community of global organizations working together. And ultimately, all of us share the same goal of, getting better treatments and quality life for our children. So that's what we're on a mission to do, and welcome feedback and thoughts and opportunities to talk with all of you. So please reach out anytime. And our website has a ton of information, and you can reach any of us through that. And I'll just open it one more time to see if there are any other questions before we sign off.
Yeah, I've just dropped my email in the chat. It was on the last slide of the presentation, but I just dropped it there as well. If anyone wants to contact me via email.
Yeah, and any of us you can reach with our first name at
Mhm.
scnalliance.org. So terrific. Well, thank you all so much. You'll be seeing a lot more of Raquel, and I hope you all have a fantastic, not too hot summer, and we'll talk again soon.
All right. Bye. Thank you.
Everyone.
Thank you.
Bye.
Bye.